morning, class 12 students. I would like to welcome all of you in the digital class session of biology. Now, today I am going to start a new chapter that is included in your syllabus, and the name of the chapter is Improvement in Food Production. So, please make sure that you are paying attention in class in order to get the concepts absolutely clear. So, let's start. Plant breeding. Plant breeding is a branch of applied botany which deals with the improvement of agricultural crops, production of new crop varieties by bringing about improvement in their genetic pattern, supported by genetic variation and selection procedure, resulting in genetic adjustment to the physical, biological, technological, economic, and social components of the environment. There are some aims and objectives of plant breeding. The aim of plant breeding is to develop a variety which combines as many as of the desirable and beneficial characters of economic value as possible, like increase in yield. Only high yielding varieties are selected from the newly developed mutant or hybrid varieties. Higher yield of plant products like grain, fiber, fodder, oil, timber, fruits, etc. has been achieved. Improved quality regarding size, shape, color, food value, taste, etc. in food, grains, vegetables and fruits is improved. The color, size, vitamin and juice content in mango is higher sugar content in sugarcane, and protein content in pulses, fine fiber in cotton, etc. can all be obtained by plant breeding. Improved structure, agronomic characteristics such as plant height, tillering, branching, erecting or trailing habit, etc. are improved. Resistance to diseases and insect pests, viruses, bacteria, fungi and nematodes produce diseases in plants. Genotype of some varieties of plants provide them resistant to such pathogens and other pests or parasite. Such strains of crop plants are called resistant strain. Adaptability to new regions and climate, drought-resistant varieties may be developed by changing the genotype of plants. Change in the duration of maturity, early maturity and early harvest save much of labor and care. Elimination of harmful and toxic substances. Plants that produce harmful metabolites causing harm to man and animal can be reduced. Development of varieties for saline soil. Saline soils are generally less fertile. Hybrid plants may be obtained that can tolerate salinity. It will result in increased use of agricultural land. Sensitivity to light and temperature. It has been possible to obtain varieties of rice and wheat plants in which their light and temperature requirement is diminished. As a result, their cultivation is possible in a region with less available light and temperature. Methods of plant breeding. Plant breeding started in the beginning of 20th century after the rediscovery of Mendel's work. Today, plant breeding has become a specialized technology used on genetics. In India, plant breeding is actively carried out at Indian Agricultural Research Institute, Delhi, Central Potato Research Institute, Cuttack, Sugarcane Research Institute, Coimbatore, and at number of agricultural universities and research stations. Now, crop improvement has following aspects like plant introduction, selection, hybridization, polyploid breeding, mutation breeding, heterozygous gene technology. Plant introduction. The process of introducing new plants from their growing place to a new locality with different climates is termed as plant introduction. The adjustment of such plants to their new locality is called acclimatization. The new crops or the new varieties may be introduced in form of seeds or cuttings. Now, all the introduction are carefully examined for the presence of harmful materials like weeds, insects, plant, pest and other disease-causing organism. This process is termed as quarantine. Quarantine is the prohibition of entry of plants or plant material in the country having infectious disease. Now, there are basically two types of plant introduction: primary introduction, secondary introduction. Now, primary introduction means when a selected new variety properly developed in a new environmental condition and it's economically cultivated without changing the genotype. It's known as primary introduction. Like the example can be a drop variety of wheat. The name is Sonora 64, drop variety of rice, which is IR8. Secondary introduction, when a selected variety is hybridized with a local variety and incorporation of new characters takes place, then it is used in agricultural purpose. This process is known as secondary introduction, like your Kalyan Sona or Sonalika, okay, which are the varieties of wheat. Now there is a particular procedure of performing plant introduction, like the first step is procurement of plant or germplasm, okay. Germplasm mainly collected from your National Bureau of Plant Genetic Resources or from International Bureau of Plant Genetic Resources. Okay. Then packing and discharge, the second stage. Third stage is plant quarantine. Fourth stage is cataloging. Catalog will be made on the basis of the origin of plants. Okay. Like I'll give you an example. Like in case of exotic plant, EC is the type of abbreviation, or in case of indigenous plant, IC is the type of abbreviation. So EC stands for exotic collection, IC stands for indigenous collection. Okay, and in case of indigenous wild plant, IW type of abbreviation has been added before their name. Okay, and last stages or the last procedure is multiplication and distribution. This would be the final stage where multiplication and distribution is done in a nearby area of selected plants. Okay, now the next aspect is selection. Now selection is an important step in all breeding experiment which has been practiced by man since the early days of agriculture. The selection involves picking up the better ones out of the entire crop plants. The selected plants are basically separated from the inferior ones and are favored by reproducing them under controlled condition. Okay, now there may be of two main types, like natural selection and artificial selection. Natural selection, this type of selection is a normal rule in the nature and has resulted in evolution. Like Charles Darwin and A.R. Wallace have explained natural selection, and according to the concept of natural selection, the fittest can survive and the rest will wipe out. Means rest who cannot survive will get wiped out. Artificial selection, it means choosing of certain superior individual plants for the purpose of having better crops from a mixed population in which individuals differ in characters. Okay. Now there are three patterns of artificial selection, like mass selection, pure line selection, clonal selection. Mass selection means it's the most common and old method of selection. It is a procedure of selecting a number of plants, heads of seeds, phenotypically superior in desired traits from the field population, harvesting and bulking their products together for sowing the next year's crop. This process repeated till the desired character are obtained uniformly. Okay. Pure line selection. The pure line selection is the progeny of a single homozygous self-pollinated plant. The single plant with desired trait or traits are selected out of the variable population in the field. Seeds from selected plants are sown in separate rows to produce a progeny by self-pollination, and desired plants are again selected from this progeny and self-pollinated. This is continued for several generations. The inferior are eliminated from each generation. Wheat varieties like Kalyan 227, then PV 18, which have developed in Ludhiana by this type of selection. Okay, so pure line selection is better than mass selection because the selected plant retained desirable characters for several generations. Okay, and then comes your clonal selection. It has been practiced in vegetatively propagated plants like sugarcane, banana, potato, etc. In this method, the best plant based on phenotypic character is selected. It is then multiplied vegetatively and supplied to the farmers. A population of plants raised from a single vegetatively propagated plant is called a clone. Okay. Hybridization. Hybridization is the technique of introducing characters of two desirable species into a single offspring, that is hybrid, by means of artificial pollination, which to create a new genetic combination. Hybrids are first generation crosses between genetically different parents. Hybrids differ in their vigor, growth, size and yield. As a result of hybridization, hybrid varieties of cereals, oil, seeds, pulses, sugar beet, onion, tomato, and fruits have been developed. Now, depending upon the parents involved, hybridization may be of following types, like intravarietal hybridization, that is cross between two different plants of same variety. Intervarietal or intraspecific hybridization, that is a cross between two different varieties of the same species. Interspecific hybridization, cross between two different species of the same genus. Intergeneric hybridization, that is a cross between two different genera. Introgressive hybridization, that is by back crossing system, some desired gene of one species may be transferred into the genotype of another species. And the next one is transgressive hybridization. Desirable genes from two different parents are basically introduced to form an offspring plant with completely separate characteristic features. Techniques of hybridization. Now, it generally involves the following steps, like the first one is selection and isolation of plants. First, the plants to be used as male and female are selected. These plants are induced to flower at the same time. Crossing is done in a greenhouse under controlled conditions. In case of cross-pollinated plants, isolation must be done very carefully. Then comes your emasculation. The removal of anthers from the female parents before their dehiscence is called emasculation. When two parent plants have bisexual flowers, the stamens of female plant are removed in the bud stage. This is to prevent cross-pollination. Process of emasculation differs in different plants. Some common methods are like forceps or scissor method. It consists in opening of flower and removing of the stamens, which can be done easily by using a pair of forceps or scissor. Second one is hot or cold water and alcohol emasculation. In plants having small sized flowers such as rice, Oryza sativa, sorghum vulgare, and bajra, Penicetum typhoides, etc. Emasculation is done by dipping the panicles in hot water having a desired temperature for a definite period. Similarly, cold water or alcohol emasculation is carried out. Male sterility method in some self-pollinated plants such as barley, sorghum, onion, bajra, emasculation operation may be eliminated by the use of male sterile plants which have sterile anthers. Male sterility is created by manipulation of such genes. Male sterility can also be induced by spraying some chemicals like 2,4-D, naphthalene acetic acid, malic hydrazide, and triiodobenzoic acid. Okay. Bagging. The flowers on female plants are kept in isolation by enclosing them in bags so that no foreign pollens may fall on stigma. This process is only called bagging. Paper or polythene bag or muslin clothing are used for this purpose. Crossing bags are opened at the time when the act of pollination is to be performed. After that, the female cross-pollinated flower is again bagged. If the plants are dioecious, the male plants are completely eliminated from the vicinity of the female plants, which are to be cross-pollinated by the pollens of a desired species. Pollination is done by brushing or dusting collected pollen grains over the receptive stigma. Labeling. The crossed flowers are properly tagged and labeled. The labeling is done either on the bag itself or on the labels specially designed for this purpose. The labeling should have the following information like date of emasculation, date of crossing, details of parents, male and female. Next is harvesting of crop and collection of hybrid seed. So the bags are removed and the crossed heads of desirable characters are harvested and collected with their attached labels separately in envelopes. After complete drying, they are threshed individually and preserved as such. In the coming season, these seeds are sown separately to raise the F1 generation. The plants of the F1 generation are progenies of crossed seeds and therefore are hybrids. Now, let's move into post-hybridization selection procedure. The seeds of the first filial generation and the subsequent generation are then collected by different selection methods. These methods are different for self and cross-pollinated crops. There are two methods adopted for self-pollinated crops. Okay. Like the first one is pedigree method. The individual plants are selected from the first filial generation population on the basis of desired characters. These plants are harvested, threshed separately, and sown in separate rows in the next year to raise the second filial generation. The plant possessing desirable characters and found disease resistant are harvested and threshed separately, and the seeds of each plant is sown in a separate row in the next year to raise the third filial generation. So this process continued up to third filial generation. At sixth filial generation, that is F6, due to successive self-pollination, most of the lines become homozygous and fairly uniform. The plant uniform in desired characters are harvested and bulked together to constitute a variety. So these varieties are then tested and given field trials. The seeds of superior variety thus obtained are multiplied and distributed to farmers. The whole process generally takes about 10 to 13 years. Okay. Bulk method. This system of breeding differs from the pedigree method. In this case, the F3 generation plants are not maintained separately but are bulked together to form a single F3 population. In F3, that is your third filial, again the suitable plants are selected, collected and bulked together. This bulking is done for sixth generation, and in F6 the desired individual are selected and harvested separately. The products of each plant is kept separate and carried out under comparative trials. So the best performers are released as new varieties. Okay. So bulk method is simple, convenient and inexpensive method. It takes more time but minimizes the labor of the as the plants are not paid any individual attention. Okay. Now let's focus on polyploid breeding or polyploidy. Polyploid cells and organisms are those containing more than two paired homologous sets of chromosomes. Most eukaryotic species are diploid, meaning they have two sets of chromosomes, one set inherited from each parent. However, polyploidy is found in some organisms and is especially common in plants. Polyploidy can be induced in plants and cell cultures by some chemicals. The best known is colchicine, which can result in chromosome doubling, though its use may have other less obvious consequences as well. Okay. Oryzalin also will double the existing chromosome content. Polyploidy in plants. Most of the plants are diploid, that is, they possess two sets of chromosome. The plant which contains more than two complete sets of chromosome is called polyploidy. Depending upon the number of chromosome sets, the individual are given different names: monoploids, diploids, triploids, tetraploids, pentaploids and hexaploids. Polyploids are characterized by gigantism or increase in cell size and hence organ size and thus overall size. These polyploids are used in crop improvement. For example, triploids are present naturally in different crop plants. An autotriploid variety of tea in India, that is an example. The other example of triploids include seedless banana, watermelon, apple, pear, etc. Okay, so polyploidy is a pervasive in plants, and some estimates suggest that 30 to 80% of living plant species are polyploid, and many lineages show evidence of ancient polyploidy, that is paleopolyploidy, in their genomes. Polyploid crops. The induction of polyploidy is a common technique to overcome the sterility of a hybrid species during plant breeding. For example, triticale is the hybrid of wheat, that is Triticum turgidum, and rye, that is Sicale cereale. It combines sought after characteristics of the parent, but the initial hybrids are sterile. After polyploidization, the hybrid becomes fertile and can be further propagated to become triticale. Okay. In some situations, polyploid crops are preferred because they are sterile. Now, some example of polyploid crops like triploid crops, apple, banana; tetraploid crops, apple, macaroni, wheat. Okay, that is also known as macaroni wheat. Hexaploid crops like chrysanthemum, bread; octoploid crops, strawberry, dahlia. Okay. Now there are some applications of polyploid breeding, like production of apomictic crops. Now apomixis, you know the process which I taught you earlier also. Apomixis provides another avenue for the use of polyploids in breeding. Apomixis provides an avenue for the production of seeds asexually through parthenogenesis. Most apomictic plants are polyploid, but most polyploid plants are not apomictic. So in plants capable of both sexual and asexual reproduction, polyploidy promotes the latter. Disease resistance through aneuploidy. Aneuploidy has been applied in breeding to develop disease-resistant plants through the addition of an extra chromosome into the progeny genome. An example is the transfer of leaf rust resistance to Triticum aestivum from Agalops umbellata through backcrossing. Okay. Seedless fruits. The seedless trait of triploids has been desirable, especially in fruits. Commercial use of triploid crop fruits can be used in crops such as watermelons and are produced artificially by first developing tetraploids, which are then crossed with diploid watermelon. So in order to set fruit, the triploid watermelon is crossed with a desirable diploid pollen donor. Next is bridge crossing. Another breeding strategy that utilizes the reproductive superiority of polyploids is bridge crossing. When sexual incompatibilities between two species are due to ploidy levels, transitional crosses can be carried out, followed by chromosome doubling to produce fertile bridge hybrids. This method has been used to breed for superior tall fescue grasses and tall fescue by using meadowgrass as a bridge species. The same principle has been applied in fixing heterozygosity in hybrids by doubling the chromosome in the superior progeny. Next is industrial application of polyploidy. Now, chromosome doubling is reported to have an apparent effect on many physiological properties of a plant. The most discernible of this has been the increase in secondary as well as primary metabolism. The resulting increase in secondary metabolites in some cases by 100% after chromosome doubling has been widely exploited in the breeding of narcotic plants such as cannabis, datura and atropa. So in vitro secondary metabolite production system that exploit polyploidism have also been developed. So the production of the antimalarial sesquiterpene artemisinin has been enhanced sixfold by inducing tetraploid. Okay, so that is known as antimalarial sesquiterpene artemisinin. Okay, so this has been enhanced sixfold by inducing tetraploids of the wild diploid Artemisia annua. Okay, now let's focus on mutation breeding. Mutation breeding is a process of exposing seeds to chemicals or radiation in order to generate mutants with desirable traits to be bred and other cultivars. Plants created using mutagenesis are sometimes called mutagenic plants or mutagenic seeds. Okay, so crop plant account for 75% of released mutagenic species, with the remaining 25% ornamental or decorative plants. So however, it is unclear how many of these varieties are currently used in agricultural production around the world, as these seeds are not always identified or labeled as being mutagenic or having a mutagenic provenance. Okay, now there are mutagenic varieties like in Japan it's Osa Gold pear. Okay, Osa Gold pear. United States it was like Rio Star grapefruit. Okay, People's Republic of China like Purple Orchard 3 sweet potato. Okay, then Zefu 802. So there are many like this. In Thailand you have, in United Kingdom, Pakistan. Okay, Pakistan it's been termed as Basmati 370, that is a short height rice mutant. India it's like PNR 381 rice. Then Sharbati Sonora wheat. Okay, so these are some examples of these mutagenic varieties. Okay, it's mentioned according to the different places, so just go through them. Now then comes your heterosis. Heterosis refers to the phenomenon that progeny of diverse varieties of a species or crosses between species exhibit greater biomass, speed of development, and fertility than both parents. The superiority of the hybrids over the parents is known as heterosis or hybrid vigor. It is manifested in various forms such as vigorous growth, higher yield, greater resistance to disease and pest, early flowering, and greater length of life. Several varieties of plants exhibit hybrid vigor. Hybrid corn is an excellent illustration of hybrid vigor, its yield being 2 or 3 times than that of the pure varieties. Hybrid vigor has also been exploited in the improvement of sorghum, sugar beet, grasses, tomatoes, onions, squash, cucumber, tobacco, and several other ornamental and cereal plants. Okay. Now, there are some effects and use of heterosis in plant breeding, like increased yield, increase in reproductive ability, increase in the size and general vigor, better quality, earlier flowering and maturity, greater resistance to disease and pest, greater adaptability, faster growth rate, increase in number of plant plots. So all these are different uses and effects of heterosis in plant breeding. So another prominent use of heterosis in plant breeding is that it's been exploited through the development of hybrid. So it is commercially exploited in seed production of cross-pollinated crops like jawar, maize, bajra, onion, cucumber, beets. It has also been used in some self-pollinated species such as rice, wheat, tomato, and brinjal, etc. Okay. Then comes your gene technology. The improvement of crops with the use of genetics has been occurring for years. Traditionally, crop improvement was accomplished by selecting the best looking plants or seed and saving them to plant for the next year's crop. Okay, so plant breeding is an important tool that has limitation. First, breeding can only be done between two plants that can sexually mate with each other. These limits the new traits that can be added to those that already exist in that species. And second, when plants are mated, many traits are transferred along with the trait of interest, including traits with undesirable effect on yield potential. So genetic engineering is a new type of genetic modification. It is a powerful addition of a foreign gene or a gene to the genome of an organism. A gene holds information that will give the organism a trait. So genetic engineering is not bound by the limitations of traditional plant breeding. So there are different steps, like therefore the traits from any living organism can be transferred into a plant. This method is also more specific in the fact that a single trait can be added to a plant. Like it's been performed in step. The first step is DNA extraction. So the process of genetic engineering requires the successful completion of a series of 5 steps. DNA extraction is the first step in the genetic engineering process. In order to work with the DNA, scientists must extract it from the desired organism. A sample of an organism containing the gene of interest is taken through a series of steps to remove the DNA. Second step is gene cloning. The second step of the genetic engineering process is gene cloning. During DNA extraction, all of the DNA from the organism is extracted at once. Scientists use gene cloning to separate the single gene of interest from the rest of the genes extracted and make thousands of copies of it. Gene design. Once a gene has been cloned, genetic engineers begin the third step, designing the gene that will be able to work inside a different organism. This is done in a test tube by cutting the gene apart with enzyme and replacing with gene regions that have been separated. Transformation. The modified gene is now ready for the fourth step, that is the process of transformation or gene insertion. Since plants have millions of cells, it would be impossible to insert a copy of the transgene into every cell. Therefore, tissue culture is used to propagate masses of undifferentiated plant cell called callus. These are the cells to which new transgene will be added. And the fifth step is backcross breeding. The fifth and the final part of producing a genetically engineered crop is backcross breeding. Transgenic plants are crossed with elite breeding lines using traditional plant breeding methods to combine the desired traits of elite parents and the transgene into a single line. So thus the offspring are repeatedly crossed back to the elite line to obtain a high yielding transgenic line. So the result will be plant with a yield of potential close to current hybrids that expresses the trait encoded by the new transgene. So the process of plant genetic engineering is basically the same for any plant. The length of the time required to complete all the 5 steps starts from start to finish varies depending upon the gene, crop species, available resources, and regulatory approval. So it can take anywhere from 6 to 15 plus years, more than 6 to 15 years, before a new transgenic hybrid is ready for release and to be grown in production fields. Okay. Tissue culture. The technique of an in vitro culture of plant cell and tissue on nutrient media in glass containers like test tube, conical flask or petri dish under aseptic condition is called plant cell and tissue culture. Plant tissue culture is a process by means of which small species of living tissues, that is explants, growing cells, are separated from a living organism and grown under aseptic conditions, that is free from bacteria, viruses and other microorganisms for indefinite controlled periods on a semi-defined or defined medium rich in nutrients. Okay. Now there are different types of tissue culture. Aseptic culture of plants may be of following types, like organ culture, isolated plant organ, for example root tip, stem tip, immature parts of flower, fruits are cultured. Embryo culture, where isolated mature or immature embryos are cultured. Callus or tissue culture, where tissues from plant organs are cultured on solid medium. Suspension culture or cell culture deals with in vitro culture of isolated cells growing in liquid medium. It is presumed that in plant culture, heterotrophic nutrition has been noted, while the majority of intact plants are autotrophic by nature. So what are the equipment that is necessary for culture? A well-equipped laboratory is essential for such culture. The equipments include like culture vessels made up of borosilicate glass having Pyrex of Monarch's trademarks, kept in dust-proof drawer or cupboard, graduated pipettes, measuring cylinder, conical flask, beaker, filter, funnel, petri dish, okay, an autoclave for melting agar and sterilizing distilled water, culture vessels, a gas or spirit burner for flame sterilization, incubator, carry out all aseptic manipulation and transfer, scalpel, a pair of scissors, a forceps. Okay. Now culture media of tissue culture generally include inorganic nutrients like inorganic micro and macronutrients. Macro elements including your nitrogen, phosphorus, sulfur, potassium, magnesium, and micro is iron, manganese, zinc, molybdenum. Organic nutrients like vitamins, amino acids, coconut milk, yeast, malt extracts. Now vitamins like thiamine, nicotinic acid, pyridoxine, calcium, pantothenate. Amino acids include L-glutamine, L-asparagine, L-glycine, L-arginine, okay, L-cysteine, and other organic supplements are like cysteine, hydrolysate, coconut milk, yeast, malt extracts. Carbon sources like sucrose, galactose, growth regulators like auxin, cytokinin, gibberellin, abscisic acid. Okay, gelling agents like agar, a polysaccharide obtained from seaweed, is used to prepare semi-solid or solid tissue culture medium. Aeration provided by shaking culture vessel and automatic shaker. Tissue culture requires complete aseptic environment, otherwise they may be contaminated by bacteria and fungi, leading to increased toxicity in media. Sterilization of nutrient media is necessary. Tissue and cell culture helps in studying plant morphogenesis, single cell cloning, cell suspension culture, organogenesis, embryogenesis, and callus is possible through tissue culture technique. Suspension cultures grow in large scale, provide excellent system for physiological and biochemical projects. Homozygous plant is essential for breeding programs which are obtained from haploid plantlets. Propagation of valuable plants is possible from plantlets in callus culture and explants. Application of tissue culture technique in plant pathology is employed in study of tumor diseases. Tissue culture methods are being used to free vegetatively propagated plants of debilitating virus. Okay, cell suspension culture are used in the biosynthesis and metabolism of steroids and cardiac glycosides. Cell and tissue culture serve as an important biotechnological tool in agriculture, horticulture, forestry, and industry. So protoplast culture had some benefits such as intraspecific, interspecific, and intergeneric protoplast fusion could be done. Beneficial genes of blue-green algae, bacteria, and virus could be uptaken by protoplast. Genetic information could be easily transferred to isolated protoplast. So there are certain importance of tissue culture. Okay, now study of morphogenesis have resulted in improvement of tissue culture techniques. Modern techniques like single cell cloning, cell suspension, culture techniques, organogenesis and embryos development have been possible after the discovery of different aspects of morphogenesis. Plant tissue culture has thrown light in understanding of plant development and biochemical composition and genetic structure of plant cell. Using tissue culture technique, callus and apical meristem of small plantlets are used as stock in vegetative reproduction, thereby facilitating the plant breeding process. So this technique is basically used in the production of plantlets of orchids in large scale. Single cell protein. Normal agricultural production of cereal, pulses, green vegetables, legumes, etc. in our country is not quite sufficient to fulfill the required demand of food, as the growth rate of human and animal population is gradually increasing enormously day by day in our country. So the alternate sources of proteins for animals and human nutrition, which is obtained from microorganisms such as bacteria, yeast and other fungus and algae, is known as single cell protein. According to the views of modern scientists, this will meet the worldwide food protein deficiency to certain extent. To increase the source of protein, basically microbes are very useful. They can be grown very easily without any difficulty on an industrial scale. One important microbe is spirulina plantensis, which can be grown very easily on materials like waste water coming from potato processing plants, straw, molasses, animal manure and even sewage. So by utilizing this process, a huge quantities of spirulina can be produced, which ultimately serve as food rich in protein, mineral, fats, carbohydrate and vitamin. Environmental pollution can also be checked by the above utilization process. Spirulina possess high nutritive value due to the presence of vitamin A, vitamin C, calcium, iron, protein, vitamin B12, essential fatty acid, gamma linolenic acid. It increases the strength of the body and lifespan. Biofortification. Biofortification means action of strengthening breeding of crops in the field to get higher protein, oil, vitamins, micronutrients and mineral content, and also to get much better nutritional healthy quality crops, so that human beings may be benefited out of this scientific practice as done by plant breeders. And in this aspect, a great role is also played by the agriculturist for improved quality. So the most important practical methods to improvise the health condition of general public is to breed crops with sufficient quantities of vitamins and minerals or higher qualities of protein and a very satisfactory amount of fats. What are the objectives? For improved nutritional value of crop breeding technique is undertaken considering the following objectives, like the content of protein, maintaining the standard of the quality, taken as a whole the content of oil and maintaining its standard quality, to increase the minimum content of vitamin, finally overall increase in the mineral content and in addition micronutrients. Now I'll give you an example, like hybrids of maize raised approximately 2 years before contain twice the total amount of amino acid like lysine, tryptophan as compared to previous maize hybrids. Atlas 66, a variety of wheat possessing high protein content, has been used up as a donor for overall improvement of the quality of wheat which are cultivated as a good hybrid variety. Okay. Now by means of genetic engineering, golden rice has been produced where beta carotene is present in high amounts, which reduces the vitamin A deficiency. Biofortification of zinc, iron, etc. has been done in rice wheat. Okay. Animal husbandry. So a branch of agriculture that deals with breeding, care and management of livestock, whose primary function is to provide meat and milk. So therefore animal husbandry is concerned with feeding, breeding, housing and health care of livestock for getting maximum benefits of human necessity. Now, livestock includes domesticated farm animals such as cow, buffalo, sheep, etc., reared for the purpose of getting meat and milk. Meat animals are cow, buffalo, goats, and sheep, and the milk animal includes cow, buffaloes, etc. So, beside meat and milk, some animals are domesticated in farm for multiple purposes. These animals include horses, camel, bulls, deers, yaks, and poultry birds. Fisheries also provide important source of animal food concerned with rearing, catching, and selling of fish, mollusks. The practice of apiculture as beekeeping is another source of food as honey. So, role of animal husbandry in human welfare. Animal husbandry can be used to obtain a lot of benefits for mankind, like dairy products, milk, and other dairy products such as yogurt, cheese, paneer, butter, ice cream, etc. could be obtained. Meat, the demand of protein is mainly covered by cattle and other animals. Land management, grazing by livestock, can be used to control the growth of weeds in the agricultural field. Labor, horses, donkeys, yaks, etc. can be used for mechanical carriers. Fiber, sheep provides fur or wool for textile industry. Fertilizer, cow dung can be used as manure of the crop field. Blood and bones of animals are also used as fertilizers. Okay. So for today it is this much. Now it's your responsibility to go through this presentation properly. If you have any problem or query regarding this topic or any other topic that is included in your syllabus, then please feel free to communicate with me. So my mail ID has been provided at the end of the presentation. So if you have any problem, you can communicate with me in that given mail ID. So thank you and have a great day.